As we discussed earlier, the objective is to produce ATP, except in this case we will not be using oxygen. There are two main steps here, glycolysis and then fermentation. Before that though, we eat, and then that food gets broken down in our digestive system. And just to simplify things, we're going to say for now that at the end of the digestive pathway is exclusively glucose, as if we were on a constant IV drip of it or something. So glucose will be the starting point for glycolysis. A molecule called NAD+ and glucose enter glycolysis as reactants. The products on the other side we see are 2-ATP, 2-pyruvate, and NADH. So looking back, the NAD+ received a hydrogen and a couple electrons and now became NADH. Meanwhile, one glucose molecule was broken down into 2 pyruvates. The energy released in breaking down glucose was used to produce those ATP energy storage molecules. Next comes fermentation. In glycolysis, we produced our 2 ATPs, but now we have these pyruvates and NADH laying around. Normally, we'd want to shuttle these into the mitochondria for the TCA cycle and electron transport chain, but if our bodies are relying on our anaerobic pathways for ATP production, that means we have very little oxygen available and the aerobic pathway is all backed up, meaning we have to use fermentation to prevent these pyruvates and NADHs from building up anymore. So we push pyruvate and NADH through fermentation, and on the other side get carbon dioxide, abbreviated here as CO2, ethanol in yeast, or lactic acid in humans, and last but certainly not least, NAD+, and we'll talk about why on the next slide. From fermentation, we produce NAD+, and this is key. Remember, on that second slide when I told you to look out for stuff, this is one of those things. The whole purpose of doing fermentation is to produce more NAD+. Why? Because if we have more NAD+, then we can package it with glucose and run them through glycolysis to make more ATP. Those 2 ATP will never get produced if NAD+ isn't available. Alright, so what about ethanol and carbon dioxide? What do yeast do with them? Well, yeast see ethanol and carbon dioxide as waste and just kind of throw them in the trash, so to speak. But we humans, being the enterprising organisms we are, found another use for those, quote, waste products. We use them to make beer and all manner of other delightful, if occasionally deleterious, beverages.